What now? Are you guys suggesting now you want us to take charge, or you are happy to take charge your end?
I can try from my end.
Okay, yeah, we still have time. We've not made it visible to others, so you can take a shot and run through if you like before we start.
Yeah, I can try to open the three of them, and then share the first one.
Yep, go ahead.
Let's see.
Tak dengar.
Sebelum mana?
Wait. All right.
Yeah, let's see if I can yeah, and now I need to share my screen. This always takes some time. Okay, let's try and let's see. Share screen. And then you will have to tell me if now when I try to go into presentation mode, if it goes to presentation mode, because it might be that
it's not okay.
I do.
Okay, yes, afternoon session. Tapi cuba tengok dia punya presentation boleh nampak juga ke tak? Tapi sekarang afternoon session, yes, correct.
So do you see it now in presentation mode?
Yes, we can see. Perfecto.
Okay. Okay, so then we still have 3 minutes.
Would you like to try the others as well, or?
Yeah, I can try.
Knock yourself out. It's okay. We have a few minutes.
Yeah. Let's see. There will always be some lag, some time. But I will try to. Okay, let's try number 2. Now I'm resuming the share. Is called Birmisco.
Correct. Birmisco, correct. We see that.
And now it's in full screen mode.
Yeah, yes, it is.
Okay. Perfect. Then I can try the last one. So stop share and then new share. Share. Yeah, I see it myself, and now I can try the presentation mode and see if now you should see it in full screen also.
The revision of TD-WG audiovisual.
Yeah.
Yes.
Yeah. Okay, so I think this will work and the 3 of them will show up. So yeah, I think we are fine.
Very good. Beautiful.
Yeah. Okay, so 1 minute to go.
Are you okay? Are you warmed up?
Yeah, I guess it will be.
Sorry, 1 minute to go.
Yeah, so you just tell me when to go, when the people are online. Soon it's going to be already 2 p.m.
Okay, hang on. Before that, let me just mute myself for. Okay, sorry, Carlos, before we let others in. So I will be doing very minute housekeeping again, and I will introduce you. So in the midst of me doing that, you can put up your first presentation. And I think we'll be ready to roll like now.
Yeah.
Yeah. But so yeah, let me do the introduction, yeah?
Mm-hmm.
Okay. Team, are we ready? You can, Carlos, you can stop your slide share.
Okay.
For now.
Should
So-
work now.
Yeah, it's okay. We can see. My camera.
Kamera,
Mana ni?
kamera ke sana, kamera.
Oh pula. Tadi okey je? Ah, okey dia. Tada. Yeah.
Kan sekali. Okey dah.
Okay, are we ready to roll? Boleh. Okay, also we have Nikisha here. Hi, Nikisha. Okay, we have some streaming problems, but never mind, no problems. All right, so again, a very good afternoon, everyone. Welcome back to the second session. So we have two other speakers in line. One is Carlos from Seckenberg Museum. And then we also have Nikisha, who will speak after, from Sandpark, based in South Africa. So before I go into our speakers, I would like to remind you all on two matters. Firstly, for tomorrow's workshop, there will be slight tweaking where instead of the first session being the importance of digitization, we will first introduce the session with how to use MAQIVE, and then later on, we will actually have the importance of digitization for research and also later for community-based work. Okay, so that's one point. The second point, as a reminder, is we will again share the post-survey questions at the end of our second speaker. So please stay tuned in order to get your certification. So without further ado, I would like to introduce you to our speaker, which is Carlos Martinez. So he's a seasoned biologist and taxonomist with a rich background in myriapodology since 2005, holder of a master's in landscape ecology and nature conservation since January 2018. Carlos boasts an expertise in the ICZN diagnosis and species description. Proficient in biodiversity informatics, he navigates standards like Darwin Core for structuring specimen and observation data. With a research footprint spanning the Caribbean, the US, Europe, Carlos is multilingual, fluent in English and Spanish, and possesses knowledge of German, Latin, and Greek rules for scientific nomenclature, which is the naming of species. Currently, he serves as a guest editor for the Ocean Species Notes series, which is dedicated to unveiling new marine species. So I will let Carlos take over from here, and welcome.
Thank you, Melissa, for the introduction. I'm also very happy to be here and right now having so many memories. So since the first time we met back in 2016 in
Sweden, yes, it's all good.
Yes. And some of the things that we learned back then, we are going to see today also.
Absolutely.
So
Looking forward to it.
It's, yeah, already, what, 7 years? So it was a good investment of time and money also.
Absolutely.
Okay.
In order to foster such collaborations again. So it's good. It's timely.
Okay, thanks. So then, good afternoon, everybody. For me, it's good morning. So it's 7 a.m. in Frankfurt, Germany. And I'm very excited to be here. And let's hope that everything will run smoothly. I have 3 presentations, so if you're not scared from the first, then hopefully you will stay until the last one. And yeah, let's try to start. Let's see if we can run it. Yeah. And hopefully you can see a slideshow already in presentation mode.
Yes, it's in presentation mode.
Okay. So then I tried to structure my 3 talks in a way that they are all connected. And one thing that I wanted to say at the start of this one is that for me, the preservation of natural history collections is not just the preservation of the specimens and the curation of the specimens, but that collections are the institutions, are the people, are the specimens, and are the archives. And I'm going to start by the archives and a bit of archive preservation and what can we do with modern techniques with the treasure trove that archives are. And this first talk is called Downstream Applications of Handwritten Text Recognition in Biology. And I brought the case of the scientist I have worked the most on, who is named Carl Wilhelm Verhoef. He used to work with myriapods, so centipedes, millipedes, and also with terrestrial isopods and other groups. He published over 670 papers and books over the course of his career. So in this presentation, let's see if I manage to run the slides, hopefully. Yeah, so first a bit of history. So there was a time between 1870 and 1920 in which Germany invaded a part of nowadays France, which is called Alsace. And during those 50 years, many developments took place, in part developments regarding the universities and the museums, and they flourished during that time. And that's the case of the Zoological Museum in Strasbourg, in Alsace, where part of this story unfolded. So the curator at the time was Dr. Ludwig Doderlein, and he was corresponding with Verhoef. Ludwig Doderlein, some of you might recall, because he was very important in marine research. So he took a trip to Japan and did studies for over 2 years in Sagami Bay. So some of you who are working with marine species might recall the name Doderlein. So he was a curator there in Alsace, and he was in contact with Carl Wilhelm Verhoef. And then the correspondence of Verhoef is preserved in the museum. And then we tried to work on that correspondence. And why is that correspondence, or why are those archive important? Well, because there are the so-called Verhoef problems, problems that he created because the way he used to work. So he was an oversplitter. He attempted to name many species, many subspecies, many varieties, because he never had a formal job, and he was mostly living from selling his specimens, his collections, also his publications, so the separata that he was getting from the publishers. So basically, the more he could name, the more he could sell. So he became an oversplitter, probably in part because of economic reasons, apart from the scientific reason of discovering new species. He did an extensive use of microscopic preparations. We will see a bit of microscopic preparations in the next presentation. Then there are also difficulties in recognizing Verhoef type specimens because many were not properly labeled. They were sent to the institutions with very little data, so now only experts can recognize them. The taxonomic descriptions of the new taxa are almost exclusively in German, so it's very difficult for the international community to deal with these works. Now a little bit more easy because of the automatic translations online, like Google Translate, but it's still very difficult to get to understand his writing. Then he has this high amount of descriptions scattered across many publications, as I mentioned, about 670 publications. And then the splitting and selling of his collections to different museums created additional problems, like the body sometimes is in one museum because he sold the bodies while he kept structures that were of taxonomic significance for himself. And then when he died, those structures ended up in a different museum, mainly in Munich, in Germany. So now we have this sort of problem that the parts of specimens and part of type specimens are in two different institutions. I'm sure that there are many taxonomists among the audience today, and that you will be familiar with some of these problems, but maybe having all of them concentrated in just one person is a bit out of the common. So yeah, that was a case of Verhoeven. And that's why it was also important to study his correspondence. He never used a typewriter. He always used hand, or maybe dictated to some of his daughters. So we needed to transcribe handwritten text. And then many things have happened regarding automated text digitization in the past 4 years. The tools have developed really quick, but I wanted to direct your attention to this former project deliverable, this IceDig.eu project, and one of the deliverables was this methods for automated text digitization. And even if this was 4 years ago, is still a document worth reading and worth revisiting, especially for this work that you are doing also in Malaysia. So we use part of that work as a background, the technical background for ours. Then regarding handwritten text recognition, Verhoeven had two handwriting styles. One is called Latin Schreibschrift, and the other one is the current schrift or Sutherland schrift. The Latin is the one that we normally use, so it's the normal handwriting. But this Sutherland schrift, very few people can read today. And there is an image there of two different letters and the two different scripts, and you will have to, so you will probably be able to read the first one, so geehrter Herr Kollege, but then you will most likely not be able to read the second line, which is in the Sutherland script, and you will have then to believe me that it says sehr geehrter, just like above, Herr, just like above, and then professor. So you see how difficult can it be to read that handwriting. So nowadays, very few colleagues can read those handwritten styles. And then there are every time less and less people also with knowledge of German who read these German scripts. So the question was if it was possible to train an algorithm to transcribe these manuscripts, and if different scripts, like the Latin one and the Sutherland one, needed to be trained separately, and if we will have to aim at recognizing individual letters or individual words. So we had all these questions back in 2018 in a Congress of Myriapodology. We have a Verhoeven workshop, and these questions were in the air. And then this work happened later. I tried together with other colleagues to give reply to these needs of the community. So what we came to is that there is software to do manual and automatic handwritten text recognition. One of the best examples is Transcribus. With this software, the work can be collaborative, and there are online and desktop clients. Now is moving more to online clients only. Then algorithms don't have to be trained in recognize individual letters or words, but whole lines of text. So that is actually really cool, because then it greatly facilitates and speeds up the recognition work. And here you can see the interface of Transcribus, in which already one image in this Sutherland script was uploaded, and where the text is already transcribed and corrected on the right panel. I had learned to translate this handwriting myself, so we use for training the software, the algorithm, some of my transcriptions, and then what was left to do was the correction. Yeah. And then one thing that will be important for many documents is that sometimes we have tabular data, more or less structure, and this software is actually able to create tables and structure the information as tables and make the tables exportable. So in this case, it was species names and then the price of the female and male specimens. And the software was able to handle that. So the other thing that we find out is that with this software, the HDR models that we needed could be created, stored, and retrained. So we could create handwritten text recognition models trained to an individual, to a person. Then it was also possible to generate statistics and charts. And with small training data, we had in total 13 documents with very little text. The validation data, so this character error rate, which is an important metric in the validation data, has been as low as 5%. And why was this important is because even in the case of German guidelines for printed documents, already 5% is the recommendation for printed or typewritten documents. So reaching this threshold for handwritten documents is a really good result, and we reached that in this work we did. And well, the other thing about the software is that it has several export formats, ranging from PDF, which we'll use, to other formats that are more suited to online representation of data, like XML and TEI. And this is a view of the statistics panel. So, for example, it can count how many lines, how many of the so-called ground truth pages, so the pages that are used for training, and how many validation pages, and then it gives a metric of the error. Yeah. So this is just for you to have some idea of how the interface looks like. And it's also possible to generate these simple charts on the training character error rates and the validation character error rates, for example. Then what can be done with this sort of work? So let's say we wanted to have the handwritten documents automatically transcribed without using too much volunteer work, many hours. So we already have this. Then apart from using the information that we can now read, what can we do with that information? And this is some of the downstream applications. Is, for example, what Melissa and I were learning back in 2016 in Sweden. And it was this software, this virtual research environment called Scratchpads, which was meant to present biodiversity data online. And the software is still active, still in use. And then I have one database there, which is called Myriatrix. So it's like the matrix of the myriapods, and is aimed at storing all things about myriapods, so centipede, millipedes, and then also onychophora, so the so-called velvet worms. And the thing is that Myriatrix has, like other Scratchpads, many modules. And one of those modules is a module on biographies. So one can have biographical information about a person. And in this case, I put biographical information on Verhoef, and then link part of that information, for example, to Wikipedia. The biographies also have a tab which is called Publications. And this is very dynamic. So it's possible to record publications and correspondence inside the database in the literature module. And as long as one uses the author name consistently and there is a biography page, this publication page will be automatically updated and recreated every time. And then I added the letters and the postcards, and those can be seen here along with the publications. So, and this is how an individual entry looks like. So in this case, letter to Ludwig Dollerline, and I use a format for recording like manuscripts or letters. And so there is a rich bibliographic information associated to the manuscripts now. And if you look at the bottom, the entries can be tagged with taxonomic names. So I tag this literature entry, in this case a letter, with the names Myriapoda, Pulida, Orobinosoma, and so on. And the effect of that is that this will link to taxon pages within the same Scratchpad, so within Myriatrix. So one is able to link what we have seen so far, documents to people, and then documents to taxa. And then you can see here that one is also able to add the full text of the document to the database. Yeah. Then it's also possible to add simple HTML markup. So in one case of one letter, there was a mention to a newspaper. I found the newspaper online and I was able to add exact link to the page of the newspaper, and it was mentioned how millipedes stopped a train because there were so many. Then in another letter, there was a mention of a publication describing this happening, and the publication was also online, so I was able to add markup to link the publication. And it can also be tagged with the name that it appeared in that publication. And then the effect of that name-wise is that then this, every time that one letter or one publication is tagged with scientific name, that also automatically generates a list of literature for a given name. So in this case, I'm giving you the example of Schizophilum sabulosum, is a millipede, and then the list that is below automatic shows the entry for the newspaper. It shows two archival documents, and then it shows the actual publication. So we have three different sources of information which are linked through one name. And then there are other tools that are automatic within the platform. So there is this Refind It, which tries to search for the name automatically and then gives list of additional publications, so it enhances the discoverability of additional information for every name. And the same happens with the Biodiversity Heritage Library search. So it's integrated into the platform and tries to search for the name and then displays a list of relevant results. And then this is the case of Germany. I don't know if you have something similar in Malaysia, but in Germany, there is this so-called Calliope catalog, which is for collections of manuscripts and is part of the National Information System of Germany and of the German libraries. And within Calliope, it's possible to create links between people based on their correspondence. So this is how it looks, what they have until now regarding Verhoeven. And this is really useful when you have it at the country level as an information system, because then you can trace correspondence of one person to many people and maybe discover additional clues on collections. So hopefully you have something like that in Malaysia, or you can implement it in the future. Then something interesting for the taxonomist and nomenclaturalist is that sometimes this sort of characters could not be differentiated. So in the first line, we have the letter I, capital I, each in an ear. And in the second line, we have capital J, Jets, Jungste, and neither humans nor machines can separate those two as they were written. And then that's why we don't know what was written, if it was Julius Horvathi Verhoeven or Julius Horvathi Verhoeven. And you can see that reflected into scientific publications. You see Julius with I, and then you see the same Julius with J in publications. So this has been part of the conundrum of many people. Okay, why there were authors who were writing the same name with different spellings, and the problem maybe was not the spelling, is that the handwriting could not be differentiated for this letter. So this is another value of studying historical documents, that it can clarify this type of let's say nomenclatural and spelling conundrums. This was a really nice thing to see from the letters. And part of this work was done while I was working for BioFit. BioFit is a specialized information service for biodiversity research. It's based in Germany, in Frankfurt. It has 3 partners. One of those is the Senckenberg Frankfurt. And I encourage you to explore this platform a bit, BioFit, and go to the digital collections and see if you can do a smarter search there in the platform. And if you find something interesting or have questions, you can always ask me, even if I move from project now. And yeah, also to make a little bit of promotion to this essential infrastructure that is in the back of many things that we use now, which is called the Global Names Architecture by Dimitri Moserim. And it's, for example, integrated with the Biodiversity Heritage Library. So just something for you to mention and maybe check later. Now we are past 28, 29 minutes. So we are finishing the first presentation, and maybe we make a short pause with 3-4 minutes for questions. And yeah, I give the word to the moderator now.
Okay, so the moderator has a question from Dr. Iswandi. Or anybody from the... No, it's already on. Yes. So before we move into the 2 other presentations by Carlos, does anybody have any questions?
Just a short question, Carlos, about the HDR project. So currently you're working in one author, Verhoeven, if I pronounce it correctly. So what are with other earlier taxonomies? So does it require to do such a process in order to decipher their handwritings?
Well, it depends what one wants to do. In my case, I was trying to track type material from Verhoeven across several European museums. So then I'm also tracing his letters and archives to try to find the clues and what happened to the type material. So that's why I'm trying to do this work. But I guess as tools advance, so now we can connect this automatic handwriting And then we have machine readable text, and then we can input that text into maybe Google Translate, and then the people can read it in English or any other language. So I would say that it won't be necessary for everyone to learn these scripts, but it's a good skill and a good thing to have in mind that collections are also their archives, which is the main message of this presentation.
Okay, cool. All right, thank you.
I think, Carlos, we can also proceed with the next presentation.
Okay. Thanks.
Thank you again.
And let's try, I close this PowerPoint to have one less. Okay, now it should be visible the next PowerPoint, and I'm going to try to put it into presentation view, and then you tell me if it is visible.
Visible.
Okay.
Perfect.
Okay, so this PowerPoint is about virtual microscope slide collection. So it's a software, and for sure it's called VIRMISCO. And I brought it because you're working a lot with images, as I understand, and this software might be useful for you. It's not a work of mine, it's a work from colleagues from Senckenberg. In this case, not Senckenberg Frankfurt, but Senckenberg Görlitz, next to the border with Poland. And this work was presented in 2018 and published as part of the proceedings of the International Congress of Myriapodology in Krabi, Thailand. I went to that Congress too. And yeah, I'm going to present this publication and work now. And what is VIRMISCO? Why to develop a software for a virtual microscope slide collection? Well, I think we all have very clear that digitization allows rapid access to research objects by researchers, but also by teachers, for example. And then it's very easy sometimes to take pictures of big specimens, take pictures of maybe pin butterflies or of herbarium sheets. But for these transparent to semi-transparent three-dimensional microscopic objects, that's not so easy. So taxa such as microinvertebrates or body parts that are very small are usually less represented in databases. So then there are other problems like the mounting media for permanent microscope slides deteriorate after some years, some decades. It might be that some were preserved after 50 years. There are cases of good preservation after 150 years, but for many slides, this is not the case. And then we have either partial damage or total damage and even loss of the object, and then the slide has no more value and needs to be discarded. So then we also have the problem of the restoration of slides. So very often we don't know anymore what was the composition, even if it was the same manufacturer, even if one knows, okay, this was this sort of preservative, then the formula might have changed over time and one does not know which formula was used. So restoration is difficult because the material or the preservative is not known, and then it's always labor intensive. So there is a lot of care to be put into restoring slides. But then we do know that for these slides, and especially type material mounted in slides, we have an important and urgent need for digital preservation. And here's where this software comes into play. And one thing that I wanted to say at the very beginning is that this software is open source and can be installed by any institution. It's available on GitHub also. So if you search for Virmisco and GitHub, you will find it. And then it's maybe something for you to try for a next funding phase, and then have funding for good software developers who can maintain and expand the functionalities of this software further, and maybe help you install it in Malaysia if you're also dealing with slide collections. There is a web page. There is the citation of the web page itself, which is different from the citation from the paper. It's available. I checked yesterday with my colleagues to put it online. They were doing a maintenance, but now it's back online, so you can check it. And there are a lot of information there. So there is information on the manual and the recommendations and what they did, and then also in the terms and conditions of usage. So please also check those if you're going to use images from there. And I'm going to move on. So I have some screenshots which were also published in the paper. And the first screenshot is of the search page of the online database. So you can use several keywords, and then you also have an advanced search with, for example, geographical terms like country, province, region. You can also search by date. You can do quick filtering with, for example, like one word taxon names, or you can search for higher taxa or genera or so. So you have all the basic functionalities in the search interface. When you do a search, it takes you to a results page or interface, which also has rich data. Then you can there select a photo micrograph because there might be more than one and more than one view. You can drag the preview image online to see the selection box, and then you can also reorder the found items or delete them from the search results, and then show those that you want in the viewer. And the viewer is the actual core of the Virmisco software, and it is very important. Is very, more than important, I will say, is enjoyable. So they actually manage to give the feel of using a real microscope. So it's possible to use these let's say image files or video files to virtually focus through the several layers of the object online. And then there are other functionalities such as search and rotating and zooming. Measuring is extremely important. So many of the early descriptions might not have measurements, but as long as the slides are properly digitized with this interface, then people could do new measurements and add those characters to review with descriptions, so to read descriptions of material. And sometimes measurements are really, really important for species distinctions. And then also like changing brightness and contrast and taking snapshots. There is a function to leave feedback. And then it's also possible to download the JPEG files or the video files. But again, please check the terms and conditions of use if you're going to do this. And this is how the viewer looks like. I really, really encourage you, when you have time, to go to the platform and do a search, and then go to the viewer and then try the focusing of the virtual microscope itself. And I think you would probably like it as much as me. And then apart from this microscope feeling, you will see that there is rich metadata associated with the images. So metadata on the taxon, on the collection object, on locality, on the microphotography itself, so the camera settings and microscope settings and so on. And this is very, very important for data reusability, and the software allows to capture this data. So if this is something that you would like for yourself, that is already implemented in the software. Then the most important thing I will say from this project is the recommendations for digitization which came out of it. I think that for no one is a secret that the choice of equipment depends on many things. Some are very practical, like what does the institution need, what is the budget, and then of course which taxa and characters are being analyzed or want to be documented. Then in this case, again, we are working with slides. It's possible to achieve good results in the photography using the typical bright field microscopy. And then it's also possible to obtain good results regarding lateral contrast with this differential interference contrast microscopy. So that's something to keep in mind, that if you can have access to this technique, then you might obtain good lateral contrast for a set of images. If you want to capture the three-dimensionality as you see it in a microscope, then you need to take images at different focal planes. And then it's very important to note that typical auto-montage image or focus stacks, in this case, with transparent or semi-transparent objects, they can produce errors in the final images. So best thing will be that if it is available, that you have a digital motor focus or macro rail to take these single images, rather than auto montage or focus stacking. Do not stack these images, please. Yeah, then the other thing is that the T format is recommended as common master image file. That might be something for you already right now. That each image should have a scale bar and with a linear measurement embedded. Then you need all this metadata on the taxon, on the collection, on inventory, sex, label information. Information on the documented part or body region and the view of the object, if it's ventral, if it's lateral, that's connected to the next presentation. And then the technical information, as I already showed you in the viewer, so microscope settings, camera settings, and so on, which is very, very important for image reusability. Yeah, and then of course, it's very useful to have an overview of the full image of the complete microscope slide to give a quick impression to the users. Of course, you also need experienced taxonomists to guide the work of image acquisition so that the characters that are taxonomic relevant can be seen in the set of images, because this is not usually what happens when only one full image is captured. So this is the other thing. It's not enough with capturing the single total view image of the slide or the object. One usually needs to document specific characters so that the collection itself is useful for remote examining. And yeah, so what are the benefits of having this sort of software and digitizing the slides? Is that, well, one helps on preserving important or fragile material, in this case, the slides and the objects contained therein. One may avoid loans because, as you saw, you can obtain really good images and then you can do measurements already online. You get a digital archive for image files. You get the possibility of remote taxonomic determination by experts because the images are online. You get a reference library for both research and education. If the images are online and with proper licenses, then you get supplementary material for publications, for example, redescriptions of taxa. And then, of course, you can also integrate their images of what you send, for example, to GenBank. If you already took slide images or photograph to send to GenBank, accompanying a voucher, then these images you could also incorporate into this software. So as vouchers for molecular investigations. And that's the end of it. Maybe we have 2 minutes for one quick question before I move on to the next presentation.
Yes, Carlos, we have a question from Dr. Iswandi actually.
Hi,
Hi, Carlos. Sorry, it's me
again.
again. Just two questions, if you don't mind. One is, will it be possible? Sorry. Okay,
Talking.
Okay. Carlos, so two quick questions. One is, so in terms of slide, those slides images are made on the flat slide, or you can also make a good images from concave slide, specimen embedded on concave slides. That's first question. The second question is, hypothetically, if a type specimen destroyed for whatever reason, fire or because of the poor collection management procedure. So if we manage to capture a very good images of the type specimens, so what will happen to the images if the physical specimen, the type specimen is already destroyed? Can we still consider that as a E-type and then use it for prescription or review? Or should we assign another specimen as neotype and take good images of that neotype specimen as well? So those
Thank
are
you.
questions. Yeah.
Thank you for your questions. So basically, as long as these macro rails or like step imaging equipment is available, one could basically photograph any three-dimensional object by layers. The difference with the slides is that as they are transparent and semi-transparent, when one does this sort of image acquisition, one gets more than just the borders on focus. So one might get internal structures as eggs, for example, or genitalia or so. So that's why I see this technique more used for transparent and semi-transparent objects. But yes, technically one could take any series of vertical pictures, and then one can assemble the images as a video file, in this case, OGG video files. So yeah, it's technically possible. And the other question is, if one does this sort of very good documentation of type specimens, and the type specimen then gets destroyed, so the slides get destroyed or the entire slide collection, then it won't be needed to designate neotypes because the slides exist as digitypes. There are not types, but they exist, and as they are a good source of information and also for measurements, for example, as long as they can be used to separate species, then there is no need to designate a neotype because there won't be a doubt on the identity of the taxon. Of course, if we go to cryptic species, molecular species, then we might need neotypes. But for morphological examination, then these digital images could act as digitypes. Yeah.
Perfect.
Good.
Thanks, Carlos. I think we can go with the last presentation.
Yeah, I try to go quickly. We have
worries.
only
No
10
rush, take your
minutes
time. Not
left.
too much.
This is sort of intellectual high tea.
Okay. So now I try to again put it on presenter's view, and you tell me if it is there. So is it seen full screen?
Yes, full screen now. You can proceed, Carlos.
Perfect. Thank you. Okay, so this last part is not exactly about software. It's also a bit about software, but it's more about biodiversity informatics standards. And this is a small review that I made of two recent enhancements to this formerly called Audubon Core, now called Audiovisual Core TALWIC standard. And these two parts are called subject part vocabulary and subject orientation vocabulary. And they are important, as you will see very soon, for databases. So if you remember the previous presentation, part of the metadata that should be captured on the object was which part of the body and which view. So these two vocabularies respond to that. Then you can find the information on audiovisual core on the official page of the TADWIC, of the Biodiversity Information Standards, which interest group maintains the standard, what is the purpose of audiovisual core. So it's basically representing metadata for biodiversity, multimedia, resources, and collections, and to make it possible for users to determine whether those resources would be fit for biodiversity science application in the broad sense. So I really encourage you to visit the TADWIC page for audiovisual core and read the documentation, especially those among you who are maybe database developers, but also to scientists and technicians who are working with, for example, describing images in the broad sense, also for publications. And then there is a bit of history of the development of these two vocabularies. So the maintenance group that was formed in 2018. Then there is what is called a task group, in this case, a views task group in 2019. Then this task group generates a feature report based on the input from the community. This was done. And then it is created something that is called implementation experience report. And this was also published earlier this year, January 2023, can be found online. And the proposal for adding these controlled vocabularies to the other vocabularies that already exist in audiovisual core was ratified in April this year. So how does it look? How it was described in the implementation experience report as it's published? For example, we will have a type of organism like an insect, and then we want to have some subject part controlled vocabularies for parts of the body. And in this case, for example, we can have head, abdomen, thorax. Then for woody plants, we might want to have leaf or bark or trunk or roots. And then the parts themselves, when they are imaged, they might be in different views. So in the case, for example, of abdomen, maybe the abdomen of an ant, maybe it's photographed in dorsal view and in ventral view. And the same for a leaf, but in this case, an upper view and a lower view. And all these terms can be organized in different concept schemes. So simple knowledge organization, and that gives structure within databases to make all these terms to link together. So for developing databases and implementing this sort of vocabularies, you should always look at these SCOS collections, the definitions, the descriptions, and then to each individual scheme or vocabulary. Then how does this look like already implemented in a repository like Zenodo? Is that we can have pictures of a specimen, of a species, and then we can have words like head and anterior, which stands for anterior view of the head. The images, of course, can have metadata, and among the metadata and the custom keywords that can be then used for filtering, then we can already see subject orientation with the control term anterior and subject part with the control term head. So this is already being implemented into databases. Then I made a review of this after the report was published because I found several issues. So there were some general issues with the documentation. I'm not going to bother you with those. They are now solved across many other vocabularies. I highlighted some general limitation of this SCOS relation in the context of ontologies for the semantic web, which is another thing with which I work. That requires more than SCOS to be solved. It's very technical, but it's just for the developers among you to ring a bell maybe. I found that one subject part term was not a subject part at all. It was a life stage value. So we got the term egg as subject part, but egg is actually a life stage value and is already present in another TADWI vocabulary. Then there were several subject orientation terms that are not subject orientation, but subject part terms. There were some important subject part terms missing, like body and trunk, in the case of trunk for both animals and plants. There were some part terms definition that were too restrictive. For example, mandible was too much defined as for vertebrates or mammals. There were also counterintuitive terms like entire organism instead of body or habitus as we use it in publications. And there was important information missing in subject orientation terms, like for example, the terms ectal and mesal, which we also see in publications, are not yet in the subject orientation vocabulary. And I documented all this online, so I'm in contact with the original developers of these two vocabularies. And now I'm the main, how to say, I lost the word. So I am the one slowing down the things in the sense that I should complete my review and format it for everything they need so they then can go on with internal review, or we can all go with a review of this standard. But everything is documented online and actually available. So in the audiovisual core GitHub, so anyone can also check what I have written so far, even if it is not finished, because I like that writing online. So then everyone can see where my thoughts are going. I already have the information in advance. And I implemented a bit of this also in Miriatrix. So Miriatrix already had terms for tagging images like preparation technique, imaging technique, keywords, taxonomy. All those are terms that are already available within any scratchpads to tag images. And then I created the subject vocabulary morphology for the part vocabulary and the subject orientation for the same subject orientation. I am not testing them yet, but they have been created. So here you can see the morphology vocabulary for subject part, and then I created a sort of hierarchical structure for the body of centipedes, and then that will help to tag images if I make it available for image tagging. And the same for subject orientation. I already entered the terms that are relevant to centipede images, including these ectal view and mesal view terms that are not in the standard. So just to show you other of the possibilities of scratchpads as a software. And the take-home message of this third talk is that new TADWIG vocabularies must be reviewed before implementing them into databases, because even if they are discussed by many people, there can still be issues with them. And some of those might be critical issues, so better to check, to discuss once more before transforming those issues into code that then is or might be harder to review. And yeah, if you are writing software to tag images, then this review that I have done will be relevant to you. You can always contact me if you have questions, and then I can provide advice to the best of my knowledge. And I'm done. And I think we are only with 1 minute left.
Mira.
Carlos, there's just one short question. I think your last presentation will be very technical, especially for those newcomers in taxonomy and also in digitalization. So in terms of database, will that be possible to use the vocabularies in other databases than Sketchpacks?
Yeah, it's always possible to use them in other databases, but more than databases, it's always good to look into these vocabularies when one is writing, for example, the figure captions of images and drawings for taxonomic publications, because there is some general guidance there for some groups. So which views to write and which body parts. And if images are captioned consistently across publications by people who know this standard, then it's easier to do text mining in the future or automatic information extraction, because it's possible to go to the figure caption and then say, okay, lateral view, dorsal view, and so on, and then this body part, that body part. So one can maybe in a very short time, with the proper information extraction tools, to create an image database from freely available publications. So it's always good that taxonomists use these vocabularies correctly when they are captioning images in taxonomic publications. And of course, also editors and reviewers.
Right. I think I'll chat to you later.
Okay. Then many thanks. I have taken 2 minutes of someone else's time, so already my apologies, and many thanks for the invitation.
Okay, thank you, Mr. Carlos and Dr. Izwandi. So, hi, I'm back. Do you miss me? Okay, so without wasting any more time, we are thrilled to introduce our last speaker of the day, Miss Nikisha Singh. Our next speaker is employed by the South Africa National Park, Kruger National Park, as the curator of the invaluable Sokusa Biological Reference Collection. Her master research predominantly focused on the aspect of the biological and ecology of the South Tree Agama and through urban context. Inviting Mrs. Nikisha Singh to present her sharing session on the digital archive of aquatic organisms from South Africa Park.
Okay. Good afternoon, everyone, and good morning to those that it's still morning for. As I've been introduced, my name is Nikisha Singh, and I'm the current curator for the Sokusa Biological Reference Collection. Okay, I just want to share my screen. Okay.
Is everything okay on your end?
The Sokusa Biological Reference Collection serves as an invaluable and indispensable...
Sorry, Nikisha. We can't see your slides, and we kind of lost you. Yeah, and we lost you for a while.
Okay, let me try again.
Yeah.
Entire screen share. Okay, can you see my screen now?
Yes,
Yes.
but not
It's not presented.
not presenting mode.
No, on the slideshow.
Slide.
Okay, can you see it now?
Yes,
Yes.
yes.
Okay, perfect. Thank you so much. Okay,
No
I'll
worries.
start now. Okay, cool. All right. So just a bit of background. The Skukuza Biological Reference Collection serves as an invaluable and indispensable asset to the Kruger National Park. Okay, so the collections were established during the 1950s when the first formal plant collections were carried out by Mr. H.P. van der Skeep.
Sorry, Nikisha, I have to stop you for a while again. Can you please? Oh, okay, for our audience, I really apologize for this technicality. Bit of a problem in South Africa now with signals, but we'll get back in a short bit. So stay tuned for a while.
Function of our facility. Okay, firstly, the Skukuza Herbarium and Museum
Again,
facility.
Nikisha, so sorry we lost
Yes.
you again. Oh dear.
Kalau call dia tidak boleh.
Boleh.
Dia tak tahan.
Right, should be able to see now. Let me just check. All right, is it all right now?
Please hide.
Okay. All right, is it all right now?
Yes,
Yes.
good.
Okay.
Nikisha, just another thing is, in case we lose you again, because I think you may not realize that while you're presenting, you might lose track of us. So we might actually go to the backup. Nikisha, I apologize,
Yes.
but I think we're going to have to take the option of playing your backup. You can stay.
Yeah, I think
But-
so. Thank you so much. It looks
Yeah,
like we're having connectivity issues on and off.
No worries, we understand. Which is why we made this plan to back it up. But please stay with us so that if the audience has any questions, you definitely can answer live. Is that all right?
No problem.
Okay, so we'll
Perfect.
prepare your recorded one, yeah? So please stay with us. Thank you so much again.
No problem.
Cheers. So for those online, please stay tuned with us. We're just finding the recordings to play of Nikisha's presentation, yeah? Thank you very much.
Okay. All right. Good morning, afternoon to everyone. My name is Nikisha Singh, and I'm the current curator for the Skukuza Biological Reference Collection. The reference collection serves as an indispensable of history. The reference collection began in the 1950s with the herbarium collection. Approximately 4,000 plant specimens were collected by Mr. H.P. van der Skeep, and those specimens are still housed in the herbarium today. Faunal surveys took place between 1959 until 1970, and this was carried out by Dr. Udev Pienaar. A number of different specimens from various taxa were collected, including amphibian, reptile, bird, small mammal, insect, and other invertebrate specimens. So why do we have such a reference collection based in a national park? Okay, one of the main services that we offer is identification. So we do get requests from scientists, interested persons, the public, students, etc., of identifications needed, for example, a plant, insect, reptile, or other flora and fauna. Okay, it keeps track of the inventory of flora and fauna that occur within the Kruger National Park. If you do not know what plants and animals occur in a certain area, you aren't able to really protect it. So therefore, it's important to know exactly what occurs where, so that we can document things like population distribution, changes in population, and this can then be filtered down into management decisions or park management. Okay, we also have international and national researchers that visit the collection. They may be looking at taxonomy of a certain plant genus, or they may want to describe or look at the description of a certain species of animal, and then they're able to answer a number of questions using these specimens. And one of the most important components is education. We have many school groups and university groups that come through to visit the collection and to learn about biodiversity. Why is it important, and why do we need to preserve it, essentially in terms of collections? Okay, so the types of collections that we have in this particular reference collection. Okay, so we've got the wet collection. This comprises of specimens preserved in 70% ethanol. It ranges from amphibians to reptiles to small mammals, and even some insects. We have the dry collection, which mainly consists of the insect collection, the pressed plant specimens, and the mammal skins. Okay, just a little bit of the stats. We have approximately 10,000 insect specimens, 1,257 reptile specimens, 487 amphibian specimens, 832 bird specimens, 129 mammal specimens, and more than 950 fish specimens. Okay. Just a few pictures from the educational facilitation and outreach that we have done this year. We have had some groups come through to learn about different aspects, how herbarium specimens are collected, etc. And then we've also had the privilege of participating in the launch of the National Science Week this year, where we exhibited the different types of collections, and we spoke to high school students about the various careers that one can take in natural history collections. Okay. So I'm just going to give a little bit of input on one of our collections and how we actually go about to the field and do some collecting. One of these is the fish collections. So every year we go out during the September time to do freshwater monitoring in the Kruger National Park. This covers the 5 major river systems, and we do collect fish specimens from these different localities. Once fish specimens are collected, they are euthanized using clove oil, which is a humane way of euthanization of fish. And we use 4 to 5 mils per 5-liter bucket, and the fish stay in there for 10 to 15 minutes. Once this is done, the fish are then transferred to formalin, and after about 3 months, the fish are then transferred to water, and then different stages of ethanol until it's 70%, and then it goes into the collection. Okay. Just a few images from our field work and the different methods that we use when collecting fish. If you look at the bottom left-hand corner, we use something called electroshocking, which is basically a mechanism or a device that uses an electrical current or an electrical field to temporarily stun fish. And then this is also means used for collection. And then we also use something called a seine net, if you look on the bottom right-hand side corner. Okay. And Kruger National Park has quite a diversity of fish. I've put on some of my favorites. So if you look at the top left-hand corner, that is known as an elephant fish. Then Coptodon rendali is the red-breasted tilapia. This tilapia is actually quite interesting, is because when you do hold them, they sort of become a little bit aggressive, and the bottom part, the breast, it becomes quite red. So therefore known as red-breasted tilapia. The bottom right, left-hand side, sorry, is the buttercat fish, and on the the bottom right-hand side is the banded tilapia. Okay. I will go into a bit of the plant collection now and the process of plant collecting and what each stage entails. Okay, so we have collection of plant specimens, drying, mounting and labeling, capture of bioecological data, and then the digitization. And we'll go through each step. Okay. So with regard to plant collection, there is a certain equipment that is needed before going out into the field. Ideally, you would need a large plastic bag, or a black refuse bag can even be used. This will be to collect all your plant material. Pocket knife or secateurs, which makes it easy to cut the plant material. Notebook or plant collection sheets. These are very important to collect the bioecological data associated with the plant specimen. For example, you would want to take down habitat details. Is the plant growing in a rocky outcrop next to the road, next to the river? You would want to take down GPS coordinates. Any other details such as flower color or smell, anything that is going to fade or sort of not be visible once the plant has been collected. So these are all essential things that need to be recorded. Okay, pen or pencil, and a field or temporary plant press. Okay. Okay. And why do we do plant collections? Okay, for identification purposes, expansion of the herbarium so that we have plants occurring from different localities, inventory of bioecological data. Again, this can be distributional data, population data. We sort of have an idea of how different populations of different genuses or species are doing. Do some of them need to be monitored more regularly? Okay, and plants are collected and preserved in various stages, meaning seeds, fruit, immature stages, mature stages, all different stages can be collected, and then further research can be done on this. Okay, databases and data management. With such a large collection and all of this associated bioecological data, we do need databases in place to manage all of this. Okay, so I'm just gonna run through a little bit about our database management. Currently, for our zoological data, we are using Access databases, but the Access databases will be transferred to something called Specify, which is something that many natural history museums are currently using, and we are in the process of transferring some of our data sets and to see how it fits in and how things will operate. Botanical data is currently stored in BRAHMS 7. BRAHMS is the Botanical Records and Herbarium Management System. But we are also in the process of moving over to BRAHMS 8. Okay. Last year, we embarked on a massive herbarium data project. we currently have 16,000 botanical records, and this data set was cleaned, updated, and standardized. We had assistance from many colleagues as well, standardized in terms of taxonomy, for example, plant name changes, IUCN red listing changes. Various resources were used, such as the South African National Plant Checklist, Plants of Southern Africa, IUCN Red Checklist, etc. Okay, the future of the reference collection. Okay, the future is digitization. So what essentially is digitization? It is simply putting something in a digital format and having that available on an online platform so that it can be accessible globally. So why should we digitize collections? If we look at all the natural disasters that have been occurring far and wide, natural collections are at risk of deteriorating if exposed to fire and flooding. Digitizing a collection also ensures that the physical integrity of the specimens are intact, because if something is available digitally, it does not need to be handled as often. And one of the main benefits of having the collection digitized along with the associated information is accessibility, which is the end goal. We want it to be accessible globally, so that someone wanting to probably do some research on a specific plant or animal, they can then go onto the online platform and have a look at the description or the morphology, and they can answer many more research questions, which will increase scientific output. Okay, just a bit of our digitization workflow. We have something called a pre-digitization workflow, which fits into the main digitization workflow. Okay, so the first step would be the repair of a specimen when required. So sometimes as we're going through specimen sheets, leaves may be loose or a certain branch may be loose. This needs to be repaired, and wood glue can be used to do this. The taxonomy is updated. So if there is a genus or a species name that needs to be updated, this will be done and also updated in the database. Each specimen also needs a unique barcode. So these barcodes are placed on the specimen sheet itself, and no two barcoded specimens in the world are similar. Okay, and then the digitization workflow itself. After the pre-curation digitization, we have the imaging process, and once the images are done, these are then imported into Lightroom Classic. The images are edited according to specific standards and protocols, and then these will be exported to a hard drive and then the cloud. Okay, so just a bit about image standards. What is required when taking out an image, and for it to be up to the standard that it can be loaded onto a national and international platform. A scale bar with the logo of your institution, then a color checker is required, and a unique barcode. Okay, just some of the images of specimens that we have taken out. Okay, and some of our progress that has happened this year in relation to the digitization project. We have something called a Lightbox setup. We're basically using a Godox Lightbox with a camera stand. This ensures that the images taken are of high quality, and lighting can be changed to what is required. We've captured a lot of the missing data from specimen sheets as well. We have been able to secure equipment such as camera, and other lighting, and this project has also received awards. Okay, and just some of the pictures of the the processes. We've also had a team that is part of the NSCF, which is the Natural Sciences Collection Facility, that is basically a virtual hub of museums and herbaria or institutions holding natural collections within South Africa. The data tech team, along with our colleagues within the herbarium, have assisted with barcoding the 16,000 specimens that we have housed. And I think that has been a very big accomplishment for us. Yeah, and then what's next? So we are now in the process of starting to capture all 16,000 images. And essentially, we want to get this uploaded to GBIF with assistance from SANBI, which is the South African National Biodiversity Institute, and other platforms. And then this will be accessible to the public. Thank you.
So that's sorry about that. Okay, so that's the end of Miss Nikisha's sharing session. Personally, I have a question for Miss Nikisha. So, as we know, South Africa is one of the most biodiverse countries. But personally, I want to ask you, what do you think is underrated specimen that you think should get more highlighted on?
More highlighted. All right. Well, when it comes to big parks or when it comes to big parks in South Africa, it's the more charismatic species that get quite a bit of attention, like your lions, leopards, rhinos, etc. But when it comes to the smaller fauna, such as your insects, invertebrates, and even plants, I think a lot more attention sort of needs to be allocated to these to that aspect of biodiversity as well. And I think it just comes down to awareness, like making people aware of the other smaller critters that are also important.
Thank you. So I'm going to ask the audience if you guys have any questions. Please feel free to write in the chat box or FB Live in the comments, and we can read it out for you.
Perhaps while waiting for that, I could ask you, Nikisha, if that's okay?
No problem.
All right, so 16,000 specimens, well done. That is in itself a big feat. I mean, it's pretty much that way. So we can all relate. I mean, we all share the same pains and struggles, the blood, sweat, and tears. Yeah. So now,
definitely.
I think I may have missed a slide or so, but with regards to digitization, how have you made a lot of your work accessible to the public?
Okay, so we're still in the process of doing that bit, but once all of our images have been captured, like the herbarium sheets, we will be using an IPT through GBIF to make it accessible, because GBIF is the Global Biodiversity Institute, which is a platform that is accessible to anyone. I mean, someone sitting across the world can just make an account, log in, and search for whatever data they want, from checklists to occurrence data. I mean, yeah, I mean the options are endless. So that's our goal. But I mean, for now, before we've even had our data sort of uploaded, I do get requests from the public or scientists, etc., for information, and that's how I've made it accessible for now. Yeah.
Okay, awesome. Thanks again, Nikisha. Perhaps we don't particularly have questions right now in the chat or Facebook or YouTube. But I do some of our audience here live with us would like to ask you some questions.
No problem.
So we have one here.
Welcome to the stage. Hi, Ray.
Okay, let's come up. Okay.
Yes.
Hello, Nikisha. My name is Tyrell. I'm a third year marine biology student. So as a marine biology student, I sometimes went to my repository that near me to get a specimens for renting or borrowing. So with 16,000 specimens, that's a great feat that you guys have done. How do you keep track all of your specimens that you have been lent to borrow or renting?
Renting. Okay, so I've been in this position for 3 years now, and I have not had any requests as yet. But the database that we are going to be using, which is Brahms, does sort of have a place, or what can I say, a tracking system where you can track loans, etc. So that's how we would keep track of loans or renting out specimens. Everything would be on our database, and then we would have all of our paperwork in order, soft copies and hard copies as well, if that answers your question.
Okay, thank you, Nikisha. That answers my questions.
Nikisha, we actually have one from the audience here. From
Yes.
I hope I didn't butcher this name. Fawaz Ghaffar. Good evening. You mentioned about editing image before uploading. What considerations should be followed when editing images without compromising the accuracy and authenticity of the visual representation? I think it's a very good question.
Is a good, okay. So there's not, so we don't do in-depth editing. What we do is sort of lens correction. So if your image is sort of obscured, the lens correction on Adobe Lightroom Classic will straighten out the image. Then we do a little bit of cropping to make sure that most of the herbarium sheet is seen. And then the last thing we do is color correction. So that little color checker that I was talking about, once you have it in that image in Adobe Lightroom Classic, there's an option to color correct, and it will use that color corrector to get it to the most what can I say, to the most accurate color that the sheet itself is without editing the lighting, etc. Yeah, that's about the editing that we do. Nothing much more than that.
Thank you, Ms. Nikisha, for the sharing session just now. It was truly informative. There's a lot of steps that I didn't know I had to take during digitizing this specimen. So I'm passing the mic to Dr. Mel to brief about the workshop tomorrow.
Okay, thank you.
Okay, thanks, Nikisha, and thanks.
Thank you,
Carlos, who's still with us today. All right, then also thank you to the audience. Thank you so much for the whole day of the seminar today. It was definitely really wonderful. Before I wrap it up, I actually want to brief you in for tomorrow's session, the workshop. So we will be starting sharp at 9. There are a few little changes that we want you to take note. In our previous tentative, we actually mentioned that the participants have to be present at the RRC laboratory. This is a minor error. Actually, all of it will be online throughout from 9 onwards right up to 5. And the second comment I would like to make is that if you were to look at the tentative, the first session was supposed to be about understanding of the importance of digitization, particularly for research, followed by how to use the MAQIVE platform. So there will be a swap in this, just more that the audience will gather the flow better of what MAQIVE is about, and then how the materials on the platform can be used for both research as well as for community engagement. So that's pretty much it, not to worry, no hassle. So pretty much that's it for me. I'm actually really much looking forward to seeing you guys tomorrow. But before we wrap up, I will pass over to Dr. Izwandi. Take it away.
All right, hello everyone. Thank you for having me there again. So I'm sitting here on behalf of the Director of Finance, Professor T.S. Fazil Mahir. He unfortunately unable to appear or to join us in this last session of the seminar. By and large, he's sending his warm regards and appreciation for all the participants that joined this seminar. By and large, we receive at the moment we have about close to 100 90 participants that joined this seminar from the beginning. So the number fluctuates in the morning and the afternoon, but overall the maximum that we had at the time is close to 190 participants, which is such a good, excellent fit considering the level or the types of program that we are organizing today. So before I end our talk, that we would like to convey our appreciation again to Sustainable Ocean Alliance for the grants that they fund for this project, which allow us to organize this seminar and also for the workshop tomorrow. And I think on behalf of the, as well, that RRC is one of the key units for aquatic including marine research in UMT. So the management system, the collection data that we had at the moment is on par with the international standards. Having MAQIVE that we doing right now, thanks to SOA again, is another attempt by RRC to reach out to the potential researchers and also audience, publics, including policymakers, on the wonders and the uniqueness of Malaysia's underwater richness. So I think RRC will be soaring high and to be on the same level with other established natural history repositories, and the university will provide support as much as possible to allow RRC to grow and become respectable repositories on par or in a similar status with other repositories around the world. So I would like to say thank you very much again to all the speakers that we had since the beginning. We have the one from Japan, Nakai-san. We have also Martin Low from Singapore, Lee Kuan Yew Natural History Museum, and then followed by Carlos from Zuckerberg. And finally, we have Ms. Nikisha from South Africa. I think your knowledge and experience that you share with us is very invaluable. And I think it will be an eye-opener to all the listeners, including our students here who are participating and observing what the actual taxonomists are working and how they disseminate the knowledge to others in the field of taxonomy and systematics. So I think that's all from us today. And I think we're gonna end our session with a photo session. And I really hope that everyone that joined us till now, if you can activate or switch on your camera so that we can have a group photo, and it will be a great momentum for all of us and show to the management that how serious or how interested people are still in terms of research on diversity and systematic of marine organisms in the world. All right, everyone, let's open your camera. Let's show your faces. So these are some of the practicing taxonomists, natural history enthusiasts, and also future researcher in taxonomy. Come, let's celebrate this occasion together. Come, everyone. So I really hope those who are hiding behind the screen, please open your camera, everyone. Come on. Let's show some faces. Okay. Okay. Just snapshot whenever you're ready. Okay.
Okay.
Number two. Dah habis dah. Itu. Orang lain pula.
Next.
Tak ada dah kan?
Next page. Oh, itu tak ada.
Okay, I do hope the number remains or even better increase for tomorrow's.
Don't forget to fill up the post survey.
Yes, don't forget to fill out your post survey because that's really matters to us, for funders and also to the university to evaluate the importance of this program. Thank you.
Okay, thanks everyone. See you tomorrow.
Ciao.
Adios.
Apa tu?